Okay, thanks. So I'm Julie Murrell. I'm a manager of an R&D group at Millipore-Sigma. This is a recent acquisition that Millipore has made to acquire the tools portfolio of Sigma. And what we do is we support both basic research, translational research, and manufacturing efforts of innovator companies to get their therapies to the patients that need help. My background is I'm a classically trained cell and developmental biologist, and my graduate work was in olfaction and vision, and I did a stem cell fellowship before going on to join the company. And today what I'm going to tell you about is the work that we've done related to mesenchymal stem cells. This is one of the types of adult stem cells that has a lot of activity in clinical trial arena right now. So I'm going to tell you a little bit about the challenges of commercial scale production. This may seem really far off for a lot of the folks that are doing the initial discovery research, but it's really one of the things that you need to keep in mind when you plan for your future success. And as part of that, we can really address things through both a closed system that really encapsulates every part of the manufacturing process, and putting high-quality reagents and harvest solutions into that manufacturing system. So the reason why this is exciting, let's see if I get this right, okay, is there are a lot of clinical trials underway for gene and cellular therapies. This is a report from the Alliance for Regenerative Medicine, and you can see that there are a lot of early stage trials, which you would expect, but there are a good number of therapies that are in late-stage clinical trials. And there are several companies that have had recent phase 3 successes, which gets them onto a path of commercialization. And one of the things that's important to keep in mind is that the different cell therapy applications require different manufacturing solutions. So you can break the cell therapy field down into a lot of different categories, but two of the easiest categories to break them down into are the autologous therapies and the allogeneic therapies. And the autologous therapies are, I get sick, I go to a company that can fix my genome, so they take my cells out, they make a modification, and they put my cells back in. And so I am both the donor and the recipient of that therapy. And for MSCs, that requires millions to a billion cells. And for T-cell therapies, that requires greater than a billion cells. And here what we need is closed manufacturing. Right now, the industry has really been piecing together different options from a lot of different things, whether it's a startup in somebody's garage that's making a bioreactor, or a traditional filtration company making some of the downstream harvest solutions, and pulling all those things together and making it seamless for the operators is one of the things that's going to drive the cost efficiencies. The other thing is the high quality raw materials and faster and cheaper release testing. One of the challenges with autologous therapies is that each batch needs to have release testing done. And in many cases, the therapies are put back into the patient before the release testing is done, so they're put back into the patient's at risk. Given that it's the patient's own cells and you have as many controls as possible in the manufacturing process, this is a low risk, but these patients are also very sick. And so if they had to wait for the full release testing, they might not be here to receive those therapies. Allogeneic cell therapies are, everybody in this room is sick, but I have really great cells. And so you take the cells out of me and you expand them and you treat one person's spinal disease and another person's stroke. The cells that can be used to treat many patients have general immunomodulatory properties, and so they can be used for many, many indications. And this type of therapy is an off-the-shelf therapy, and here what you need is to be able to really manufacture at scale. So thinking about, rather than just manufacturing in a blood bag, in an incubator, on a shelf, in a lab, you need to think about ways to scale this up to something that might be the size of a person in order to make enough cells. And then here, with that many cells and the larger volumes, you also need downstream processing. But here you still do need those high-quality reagents. There's also a push in the industry to move away from the animal-derived materials that are traditionally used in cell therapy research. So in order to transition from clinical success to commercial success, we really need process innovation. And right now, we're still pretty early on that process innovation. We can manufacture these therapies for clinical use and therapeutic use, but it's cumbersome, and it's labor-intensive, and it's expensive. And so the way that we're going to get to commercial success, and when I say commercial success, I mean something that is cost-effective to manufacture and will be reimbursed by the insurance companies. In the U.S., this is a big issue. In other parts of the world where there's single-payer options, it's less of a struggle against the many, many partners, but it is still a struggle to determine how these therapies will be reimbursed. And then also the raw materials. So what we've been working on is a complete system where we have identified cell culture media and supplements, and also those alternatives for traditional animal-derived sera, and also sterility solutions. Bioreactors for the main part of the expansion. These are single-use closed reactor systems. Downstream processing to be able to recover your cells quickly so that their effect in the patient isn't compromised. And then also combining that with product quality assays. So really implementing those identity, purity, potency assays that are needed to release the product. So I'll talk about some of the things that we've been working on in our lab. And the way we work is that we use mesenchymal stem cells from both adipose and bone marrow derived tissue that we've derived ourselves in the lab, and also T cells to pull together complete manufacturing systems. And then we work with collaborators to show that these systems work on their cells as well. But getting from here to here, we did that, right? So we developed the manufacturing systems, but what else needs to be done? So really we were looking to identify a collection of reagents that were going to be the best solution moving forward, and really enabled us to transition from that clinical success to the commercial success. So we're talking about basal media, which doesn't seem like it should be a big deal. Serum alternatives, recombinant trypsin, and trypsin inhibitors are neutralizing solutions. And I'll point out that currently serum is many times sourced from bovine sources, and the traditional trypsin is derived from porcine sources. And so these are both things that folks, when you think about where monoclonal antibody manufacturers are now, they've moved completely away from these types of materials in their processes. They've moved to a completely chemically defined process. So serum replacement, one of the things is that some folks are still using a very high percentage of serum in their processes. And New Zealand and Australia are the primary suppliers of approved serum that can be used for expansion of cells that are going to be put into patients. And then we also have the regulatory concerns, but there's also a supply concern and a cost concern. So there's been some modeling done that says that with the first cellular therapy that is approved for widespread indications, such as a stroke indication or a congestive heart failure indication. If that process uses serum, it will deplete all of the available serum that is GMP grade, all of it. And so that results in a situation that is not viable for manufacturers. And so that's why folks are looking to move away from these materials, find substitutes, and really drive towards those chemically defined media. We've been using platelet lysate in our processes. This material, this raw material, has a relatively unlimited supply because it's part of the blood donation system. And it expires after just 5 to 7 days. And so there's a lot of waste of the material. And so this is taking a waste material from a process that's already approved. So when you think about it, blood donations are screened, and then those materials are already being put back into patients. This material can be processed so that it's lysed to release the cytokines and growth factors, and then used as a substitute for the serum. And so what we saw is that when we started with the DMEM with 10% FBS, if we change that over to an alpha mem with platelet lysate, we saw an increase in growth in traditional flask culture. We also transitioned this to spinner flask, which is a smaller scale stirred tank vessel. And looking at that difference between DMEM and 10% FBS versus alpha mem with 5% platelet lysate, we saw a big difference in the growth. So not only could it substitute and give you equivalent growth, but it gave you better growth. And so if you can get better growth or higher yield of cells, and granted the cells have to be the right kinds of cells, with the same cost, that improves your cost efficiencies. So we also took this into those small-scale bioreactors, so 3-liter bioreactors. They're about this large. And we saw the same difference when we switched the media formulation and removed the bovine materials from the media formulation. And we also took this into the larger scale reactor, the 50-liter bioreactor. And we saw that not only could we get a higher yield of cells, but we could get that higher yield earlier. And what this leads to is an increased utilization of your manufacturing facilities, so that you don't need as much footprint to produce the same number of cells because you're able to do your runs in a shorter amount of time. So we also looked at recombinant trypsin. And these are just kind of range-finding experiments that we did, and then we were able to substitute them into our process for the porcine trypsin that we had been using. And we found a range of parameters that were important for utilizing that recombinant trypsin. However, if you're using recombinant trypsin, trypsin needs to be neutralized in some manner. And the traditional way of neutralizing the trypsin is, of course, adding serum to serve as the substrate for the trypsin. And we've just been working very, very hard to remove bovine sera from our process. And so here what we've done is looked at a few of the plant-derived trypsin inhibitors for use in the process. And we've just looked at some growth in both flat culture and in the 3-liter bioreactor and shown that we see very good recovery over time. So when we think about growing cells in bioreactors, and what I've shown you is a variety of results of growing the cells both on flat culture and in these stirred tank vessels. When you transition to a stirred tank vessel, for adherent cells, especially stem cells, you don't want to adapt those to suspension. What you want to do is give them a substrate to grow on. And in this case, that means that the harvest isn't just as simple as saying, oh, we're going to harvest the cells. It's really a series of steps to remove the media, wash and remove the wash, add the enzyme, let that incubate, deactivate the enzyme if necessary, and then separate the cells from the microcarriers. And so this is an image of the cells on microcarriers, and really what we want to get off, we want those cells off the microcarriers in single cell suspension. We don't want a lot of aggregates there. And we want the microcarriers to be completely empty, and those should go to the trash. And then the cells need to go into either an administration bag if it's an autologous therapy, or a cryovial if it's an allogeneic therapy and can be used off the shelf. And so what we've done is implemented a protocol where we can do this whole process in the bioreactor. And this provides some really nice advantages, because the bioreactor has agitation, which helps with the detachment of the cells from the microcarriers, and it also has temperature control, which is really important for the activity of the enzyme. And so we can see, you can't really see very well, but the microcarriers, which are white, are all settled down here. We've removed that growth media, added in some PBS, drained it again, and then we add the enzyme. And so it looks very similar to this, where we're able to agitate the impeller to get a little bit of that mechanical stress to dislodge the cells. What that does is it shortens the time that we have to expose the cells to the trypsin, compared to if we didn't have any mechanical stress. You might relate this to if you're used to using T-flask. A lot of times once you add the trypsin, you let it incubate for a couple minutes, and then you bang it, right, to get the cells off. And so you're trying to mimic a lot of the workflow steps that you're using in flat culture in the bioreactor systems. And so what we then did, because Millipore is a filtration company historically, and we have a very good history of providing a range of filtration options for many indications, is that we really stepped back and used our expertise, and we said, okay, well, we've got microcarriers that are 200 microns in size, and we've got cells that are 15 to 20 microns in size. That's a really nice size separation challenge, right? If you have to separate something that's 20 microns from something that's 40 microns, that's a big size separation challenge. But separating something that's an order of magnitude difference in size, that's really, the bread and butter of size-based separation. And so what you can see here is you can retain the microcarriers and pass the cells through. And we have several donor banks. These are two of our bone marrow donor banks. And after expansion in the bioreactor, we were able to recover 95 to 100% of the cells with very high viability. And the nice thing about this process is that it's very fast, right? So when you think about traditional downstream processing in monoclonal antibodies, it can take several days. But this is a very straightforward process. It's not requiring a lot, it's not requiring hours at each step. So this filtration happens over the course of perhaps 20 minutes. And what we can see is that before filtration, so if we just took a sample of this, so after you've added the enzyme and after you've incubated, and you have this slurry that should have microcarriers and cells and liquid, if we take a sample of that and look at it under the scope, this is what it looks like. And then after filtration, if we just look downstream of that filter, we don't see any microcarriers. And here we got a 90% cell recovery with 97% viability. So one of the things to keep in mind as you transition from T-flask to multi-layer flask to multi-layer flask that might require robotics to handle is that for allogeneic therapies, even if you increase the number of these vessels, you're having to increase the number of the vessels dramatically in order to get a cell bank that would be appropriate for allogeneic therapies. And so what this is showing here is, yes, you can do this. You would require a lot of people, so lots of those folks walking down the hallway carrying the cell factories, or the robotic forklifts that are employed in the GMP environment to handle the 40 stacks. or you can look at these stirred tank bioreactor platforms and really leverage the advantage that these have, which is all about the surface area to volume ratio. With a multi-layer stack, there's a minimum volume of buffer or media that's required in order to cover each layer. And in the bioreactor, you can decrease the volume that's required even though you increase the surface area of the microcarriers that you add. And what that allows you to do is get to, one batch for the number of doses. This also decreases the amount of QC testing and release testing that's required because you only have one batch rather than having 5 batches or 25 batches. So hopefully what I've been able to show here is our research around expansion of MSCs on microcarriers, and I didn't show a lot of this data because it has been presented before. And we've taken this up to the 50-liter scale. For allogeneic cell therapies, we see that these are primary cells, right? These are not transformed, they're not immortalized, and therefore they have a limited lifespan in culture. And so a 10,000-liter tank just really isn't needed because you'd need such a long seed train that you would deplete the proliferative properties of those cells before you got to those volumes. So ranges in the 200 to 1,000 liter are probably going to be the max that you're going to see for cell therapies when they transition to bioreactors. And then we've seen that filtration technologies work well. I showed the work on the microcarrier retention so that you can recover the cells, but you can also use them for concentration of the cells into a smaller volume. And then we've also worked on animal origin-free media, supplement, and reagent systems. And so really with this and the fact that you can use the bioreactors and these scalable harvest devices, you've got this integrated cell processing solution achievable. And all of this together is going to get you that closed, scalable GMP solutions that you really need to consider early in your research activities. Thinking about identifying reagents that have both a research use only version and a GMP version that you can seamlessly transition to in the future. And so I wanted to recognize the folks that actually did the work. This is our group based in Bedford, Mass, and we have one person who's located in France as well. So that's all I have for today. Thank you very much. I'm happy to take questions.
Are there any questions?
Yes.
When you move to a serum-free system, you take out a variety of hormones that act on the beta-3 on mesenchymal, cardiac, and other stem cells, and probably make them grow. So you can still stay animal serum-free, but you might consider whether or not you could get growth to be enhanced with simple addition of a couple of factors, one of which would be thyroid hormone in low concentration.
Yes, so that's definitely something that folks are really investigating, and you see a great proliferation of xeno-free, serum-free formulations out there for growing stem cells. And what we've seen in our own hands is that those work relatively well for specific cell types in planar culture, in traditional 2D culture. But when you go to some of these other systems, whether it's a rocking bioreactor or a stirred tank bioreactor, the complexity of the proteins that's present in those animal-derived materials provides some protection to the cells. And so it's a balance between keeping the cost of the chemically defined media at a reasonable place and providing any of the shear protectants that those proteins maybe non-specifically were doing in addition to their specific mitogenic activities. So it's definitely a big balance and a challenge in the field right now.
Yeah, I was wondering, clearly you demonstrated that your trait lysase had a better supportive role with respect to sulfur as compared to SBS. Is it because of that species of used human serum? I mean, have you seen, is it possible?
So folks are using either purified HSA or recombinant HSA in some of these xeno-free formulations. And again, it works well with specific cell types in planar culture. But that transition to something that has movement or dynamic flow of the liquid does present some challenges. It's definitely something that folks have in their formulations. The recombinant HSA is just stripped, right? So HSA that's purified from human materials generally has other things bound to it, right? Even though it's purified, there are certain things that are carried along in the process.
Can I just ask you about the speed? You said it's 3 minutes. Would you like to comment on that?
3 minutes for what?
Sorry, how quickly all these things happen? 3 minutes for some processes?
So I mentioned 20 minutes for the harvest process. So being able to push all of the liquid through a harvest device to retain the microcarriers. But a lot of these things we will wash for 5 minutes, right? So it really does compress it so that you can do that whole harvest and cryopreservation in a day. So in generally less than an 8-hour shift.